In this tutorial, I will be discussing the structure of DNA and the process of replication. As you can see in this picture that you've seen before, we have here a chromosome that is only going to be visible when the cell is dividing during mitosis or meiosis. The material, when seen in a non-dividing cell, is going to be called chromatin, and that's going to be long, thin fibers that will shorten and condense to form the visible chromosomes during the division processes. And within this chromosome, we will have the DNA molecule, which is shown here, and you see the spiral shape that is associated with DNA. And this is the molecule that we are going to be talking about now in detail, understanding its structure and how it's going to be able to control the transmission of traits from parents to offspring. In this picture, you can see the molecule of DNA in its spiral shape, and on this side, you see the molecule of DNA once it has opened up in preparation for replication. Now, DNA is going to consist of building blocks. The building blocks are called nucleotides. And a nucleotide is going to have three components: a sugar, a phosphate, and a nitrogen base. Here is the phosphate molecule, and the sugar in DNA is going to be deoxyribose. Deoxyribose. Ribose is the sugar, deoxyribose, is the sugar that's going to be found in DNA. And the other molecule that we're also going to be talking about later on, RNA, you're going to be finding the sugar ribose. But for DNA, it's deoxyribose. And that's where the molecule gets its name from, deoxyribonucleic acid. So we have a sugar, a phosphate, and then a nitrogen base. The nitrogen base, there will be 4 different nitrogen bases in DNA. They are distinguished here by letters. G is for guanine, C is for cytosine, T is for thymine, and A is for adenine. Now, you have here 1, 2, 3, 4 nitrogen bases, 4 nucleotides joins together to make a chain. And how does one nucleotide join to the next? You can see here, the sugar of one will bond to the phosphate of the next. Okay? Sugar to phosphate to make what we call a polynucleotide. And in DNA, we have 2 polynucleotides. All right? Here's one polynucleotide, and here's the other one. And when the DNA is opened up, as you see in this picture, it almost looks like a ladder with the sugars and phosphates making up the sides of the ladder, or what they call the backbone of the molecule, and then the nitrogen bases making up the rungs of the ladder. Now, what's holding the 2 polynucleotides together? It's a hydrogen bond that exists between the nitrogen bases. And as you can see, they're going to bond in a specific fashion. A always bonds with T, and C always bonds with G. And the reason why they have to bond in that fashion, as you can see here, between A and T, you have 2 hydrogen bonds forming, and between C and G, there are 3 hydrogen bonds. That's why they can only bond in that particular order. And you need to memorize this in order to be able to do some of the different work that you're going to be required for this chapter. Now, the nitrogen bases can be classified into 2 groups: pyrimidines and purines. Pyrimidines, as you can see here, have just 1 carbon ring. And the 2 pyrimidines are going to be thymine and cytosine. A purine will have 2 carbon rings. And the two purines are going to be guanine and adenine. Now, we said that DNA controls the transmission of traits from parents to offspring. In order for DNA to be passed on, it has to replicate. And this replication is going to take place before mitosis and before meiosis. And it's going to happen in the nucleus because that's where the DNA and the chromosomes is going to be found. Now, it's going to start with opening up the bonds that exist between the two polynucleotide chains. Remember, the hydrogen bonds are going to be here between the nitrogen bases along the chain. And the molecule that's going to help to break that hydrogen bond is called helicase. Helicase is an enzyme. In biology, any word that ends in ase is an enzyme. So the helicase enzyme is breaking the bonds, opening up that chain. Think of it like a zipper that is closed in a jacket, and now you're unzipping it so that it's going to open up. And now you're going to have the two original strands separate from one another, one on either side. And now, free nucleotides that exist in the cytoplasm, these come from food that we eat. Food that was once living, and now when we digest the food, you're going to break it down into its components, and all of these free nucleotides are going to be in the cytoplasm and will be joining with their complementary bases. The complementary bases, remember, A bonds with T, C bonds with G. So following the sequence of bases in the original template, these free nucleotides are going to come in and bond with the aid of another enzyme, DNA polymerase. So you will have one strand going in one direction, the leading strand, and the lagging strand going in the other direction in order to complete this bonding of complementary bases. It'll continue until the process is complete. And it's called a semi-conservative process because in the end, the two new molecules that you're going to form will each have half of the molecule from the original DNA, and the other half will be the new half that's being formed during the replication process. Now in this picture, you can see the completed process. Here you have your original DNA with a specific sequence of bases. This was opened up with the help of helicase, the enzyme that breaks the bonds between the hydrogen bonds between the nitrogen bases, opens it up so that now free nucleotides that are in the cytoplasm will now come in and bond with their complementary bases with the help of DNA polymerase, and you will end up now with two new DNA molecules that are exact copies of each other and exact copies of the original strand that you started from. The sequence of bases is the same. This sequence of bases is what will be controlling the proteins that the body is going to be making. And this is what would now be going into the chromosomes that we had called during mitosis. Remember, we called them chromatids when they were held together by the centromere before the division took place. So each strand would be inside of the replicated chromatids, okay? So that when they divided, each new daughter cell would have the same genetic information as was in the original mother cell. So this completes the process of DNA replication.